Welcome to Wonderslate. Subscribe to our Telegram channel given in the description to updates and study materials. Topic for today's quiz is MCQs on cell biology. Question number 1. The term cell was given by Option A. Robert Hooke. Option B. Tatum. Option C. Schwann. Option D. They Barry also. The correct answer is Option A. Robert Hooke. Robert Hooke was the first biologist who discovered cells. Question number 2. The cell is not applied for Option A. Algae. Option B. Bacteria. Option C. Virus. Option D. Fungi. The correct answer is Option C. Virus. Viruses are not made out of cells. A single virus particle is known as a virion, and is made up of a set of genes bundled within a protective protein shell called a capsid. Question number 3. The membrane around the vacuole is known as Option A. Tonoplast. Option B. Elaioplast. Option C. Cytoplast. Option D. Amyloplast. The correct answer is Option A. Tonoplast. The tonoplast is the cytoplasmic membrane surrounding a vacuole, separating the vacuolar contents from the cell's cytoplasm. Question number 4. Microfilaments are composed of a protein called Option A. Tubulin. Option B. Actin. Option C. Myosin. Option D. Chitin. The correct answer is Option B. Actin. Actin is a globular structural protein that polymerizes in a helical fashion to form an actin filament, or microfilament. Microfilaments assist with cell movement and are made of a protein called actin. Actin works with another protein called myosin to produce muscle movements, cell division, and cytoplasmic streaming. Question number 5. A plant cell wall is mainly composed of Option A. Protein. Option B. Cellulose. Option C. Lipid. Option D. Starch. The correct answer is Option B. Cellulose. Plant cell walls are primarily made of cellulose, which is the most abundant macromolecule on Earth. Cellulose fibers are long, linear polymers of hundreds of glucose molecules. These fibers aggregate into bundles of about 40, which are called microfibrils. Microfibrils are embedded in a hydrated network of other polysaccharides. Question number 6. Glycolipids in the plasma membrane are located at Option A. Inner leaflet of the plasma membrane. Option B. Outer leaflet of the plasma membrane. Option C. Evenly distributed in the inner and outer leaflets. Option D. It varies according to cell types. The correct answer is Option B. Outer leaflet of the plasma membrane. The outer leaflet consists predominantly of phosphatidylcholine, sphingomyelin, and glycolipids, whereas the inner leaflet contains phosphatidylethylamine, phosphatidylserine, and phosphatidylenositol. Question number 7. Lysosomes are known as suicidal bags because Option A. Parasitic activity. Option B. Presence of food vacuole. Option C. Hydrolytic activity. Option D. Catalytic activity. The correct answer is Option C. Hydrolytic activity. Lysosomes work as the waste discarding structures of the cell by processing undesirable materials and degrading them, both from the exterior of the cell and waste constituents inside the cell. Lysosomes are called suicide bags because of presence of these large number of acid hydrolyses in them. Only a thin membrane separates the destructive enzymes from the rest of the cell. And if the membrane happens to get broken, various cellular constituents would undergo lysis. Question number 8. The properties of integral membrane proteins can be studied by Option A. Atomic force microscopy. Option B. Cryosectioning and electron microscopy. Option C. Freeze fracture technique and electron microscopy. Option D. All of them. The correct answer is Option C. Freeze fracture technique and electron microscopy. Freeze fracture gives a unique look at the interior of cell membranes, because it splits and separates membrane phospholipids into two opposite and complementary sheets or faces. Question number 9. Which among the following defines GPI-anchored proteins? Option A. Integral proteins of the plasma membrane. Option B. Peripheral proteins of the plasma membrane. Option C. Proteins that bind to ion-gated channels in the plasma membrane. Option D. Proteins which randomly bind to lipids of the plasma membrane. The correct answer is Option B. Peripheral proteins of the plasma membrane. Proteins bound to the plasma membrane by a GPI anchor can be readily distinguished by the use of an enzyme called phosphatidylserine-specific phospholipase. Only transmembrane proteins can function on both sides of the bilayer or transport molecules across it. Question number 10. The fluidity of the plasma membrane increases with Option A. Increase in unsaturated fatty acids in the membrane. Option B. Increase in saturated fatty acids in the membrane. Option C. Increase in glycolipid content in the membrane. Option D. Increase in phospholipid content in the membrane. The correct answer is Option A. Increase in unsaturated fatty acids in the membrane. The plasma membrane is a fluid combination of phospholipids, cholesterol, and proteins. The elbow room helps to maintain fluidity in the membrane at temperatures at which membranes with saturated fatty acid tails in their phospholipids would freeze or solidify. When we increase the amount of unsaturated fatty acids in our cell membrane, the fluidity also increases. Question number 11. Select a foodborne toxin. Option A. Botulinum toxin. Option B. Tetanus toxin. Option C. Diphtheria toxin. Option D. Cholera toxin. The correct answer is Option A. Botulinum toxin. Foodborne botulism occurs after eating food that contains the bacterium toxin, produced by C. botulinum. This toxin does not give a bad odor or taste to food. The disease most often develops after consuming improperly processed home-canned foods or home-preserved meats. Question number 12. Phosphatidylserine residues in the plasma membrane are located at Option A. Inner leaflet of the plasma membrane. Option B. Outer leaflet of the plasma membrane. Option C. Evenly distributed in the inner and outer leaflet. Option D. None. The correct answer is Option A. Inner leaflet of the plasma membrane. Phosphatidylserine, PS, is the negative phospholipid that is generally mainly localized in the inner leaflet of membranes, and its exposure is due to the action of enzymes like scrambylases. Question number 13. Which cell organelle is involved in apoptosis? Option A. Lysosome. Option B. ER. Option C. Golgi. Option D. Mitochondria. The correct answer is Option D. Mitochondria. The actin cytoskeleton and microtubules can also interact with mitochondria, 45, which are considered as the central processing organelles in most apoptotic pathways. Apoptotic signals, either from outside or inside the cell, converge to mitochondria that can initiate the execution phase of apoptosis. Question number 14. The function of the centrosome is: Option A, formation of spindle fibers. Option B, osmoregulation. Option C, secretion. Option D, protein synthesis. The correct answer is Option A, formation of spindle fibers. Centrosome function: 1. Assist in cell division during mitosis. 2. They play a role in mitosis in organizing the microtubules, ensuring that the centrosomes are distributed to each daughter cell. 3. They maintain the chromosome number during cell division. Question number 15. A cell without a cell wall is termed as: Option A, tonoplast. Option B, protoplast. Option C, symplast. Option D, apoplast. The correct answer is Option B, protoplast. A cell without a cell wall is called a protoplast. Question number 16. The oxygen and carbon dioxide crosses the plasma membrane by the process of: Option A, active diffusion. Option B, facilitated diffusion. Option C, passive diffusion. Option D, random diffusion. The correct answer is Option C, passive diffusion. The movement of molecules across a membrane without the input of energy is known as passive transport. Question number 17. The resting potential membrane is determined by: Option A, potassium ion gradient. Option B, sodium ion gradient. Option C, bicarbonate ion gradient. Option D, none. The correct answer is Option A, potassium ion gradient. The resting membrane potential is determined by the uneven distribution of ions, charged particles, between the inside and the outside of the cell. The resting membrane potential is a result of different concentrations inside and outside the cell. The difference in the number of positively charged potassium ions, K+, inside and outside the cell dominates the resting membrane potential. Question number 18. Distribution of intrinsic proteins in the plasma membrane is: Option A, random. Option B, symmetrical. Option C, asymmetrical. Option D, none. The correct answer is Option C, asymmetrical. The intrinsic proteins, as their name implies, are firmly embedded within the phospholipid bilayer. Almost all intrinsic proteins contain special amino acid sequences, generally about 20 to 24 amino acids long, that extend through the internal regions of the cell membrane. Question number 19. Which is not an example of transmembrane transport between different subcellular compartments? Option A, transport from stroma into thylakoid space. Option B, transport from the cytoplasm into the lumen of the endoplasmic reticulum. Option C, transport from endoplasmic reticulum into the Golgi complex. Option D, transport from mitochondrial intermembrane space into the mitochondrial matrix. The correct answer is Option C, transport from endoplasmic reticulum into the Golgi complex. Question number 20. Which is correct regarding the peptides in the Ramachandran? Option A, the sequence of the peptide can be deduced. Option B, It is not possible to conclude whether a peptide adopts entirely helix or entirely beta sheet conformation. Option C. Peptides that are unstructured will have all the backbone dihedral angles in the disallowed regions. Option D. The occurrence of a beta turn conformation in a peptide can be deduced. The correct answer is Option D. The occurrence of a beta turn conformation in a peptide can be deduced. Thank you for watching the video. If you'd like practice this quiz, then download the Wonderslate app now, link in the description below. If you have any suggestion for us, feel free to leave comments. If you liked the video and want to see more of it, hit the thumbs up and don't forget to click on subscribe button to get more updates of the future videos.